So, in the previous lecture, we talked about transcription in eukaryotes, specifically looking at the promoter in a typical eukaryotic class II gene or a protein coding gene. These would be, again, genes that specifically associate with RNA polymerase II, and the transcription product being a messenger RNA. We also talked about the assembly of the generalized transcription factors and the role of each of those components in the initiation and then transitioned into the elongation of transcription in eukaryotes. What this lecture is going to focus on is going to be termination of transcription in eukaryotes. That is, how is this whole process stopped? However, before we do that, I need to add a little preamble, which we will come back to in the next lecture. You should all be familiar with the fact that eukaryotic messenger RNAs undergo several processing events before they are considered mature and leave the nucleus of the cell and enter into the cytoplasm. Those three processing events are known as the addition of the poly A tail at the 3' end of the message, an addition of a 5' cap at the 5' end of the message, and splicing. So in the next lecture after this, we will discuss those processing events in a little more detail because, again, they are something that is specific to eukaryotic messenger RNAs. However, what we want to do is, again, deal with how termination of transcription occurs. And unfortunately for us, it's a complex topic because the termination of transcription is coupled with the addition of one of these processing events, the addition of the poly A tail. So polyadenylation, or the addition of the poly A tail, is a process of adding poly A nucleotides to the RNA at the 3' end. This is typically about 250 nucleotides in length and functions to add stability to the messenger RNA, as RNA molecules tend to be much more unstable compared to DNA. And they enhance translation. They also play a role in enhancing splicing and play a role in mRNA transport. We'll talk about this in a little more detail again in the next lecture when we discuss concepts associated with processing. So, as I mentioned in the previous slide, the transcription termination is coupled with the mechanism of polyadenylation. Very simply put, cleavage of the mRNA prior to termination occurs, followed by polyadenylation of the 3' end. But again, these two things are coupled or go hand in hand. So what I want to do is I want to talk about some sequence components in the gene and corresponding RNA. Then I want to outline for you or define the protein factors involved in the process. And then we'll look at putting all the pieces of the puzzle together, looking at it step by step. The first thing I want to mention are two sequence elements that occur at the end of the gene and then also obviously occur in the transcript when transcription occurs. One sequence element is known as the polyadenylation signal, and the second sequence element is upstream of this polyadenylation site and is just a GU-rich region. So two sequence elements are found in the gene and then the corresponding transcript. A upstream element upstream of the polyadenylation site, and then the polyadenylation signal site itself. The protein factors involved in this complex coupled mechanism include something known as CSTF, which is short for cleavage stimulation factor. It is an RNA binding protein and helps stabilize the complexes that form. You also have CPSF, which is short for cleavage and polyadenylation specificity factor. It is an RNA binding protein as well, and it recognizes the polyadenylation signal that has been produced. You also have CF1 and CF2, which are short for cleavage factor 1 and cleavage factor 2. It is a 2-component endonuclease that is going to be playing a role in cleaving the transcript away from the elongating polymerase. In addition, you have PAP, which is short for poly-A polymerase, which is going to be the enzyme that adds the A-containing nucleotides to the 3' end of the message. And then PABP, which is poly-A binding protein, which is a set of proteins that binds specifically to the poly-A tail. So now let's try to put all of these puzzle pieces together in a very simplistic view, and then in the next slide we will look at it in a little more detail. The first step, or the first thing that occurs, is that CPSF and CSTF bind to the C-terminal domain of the RNA polymerase 2. So at the very top of this figure, you can see those two factors bound to the tail. And what happens is the polymerase transcribes past the two signal sequences I mentioned previously, the polyadenylation signal plus the upstream site, upstream of the polyadenylation signal. So the polymerase transcribes past that site. Once that sequence element is in the transcript, these factors, both CPSF and CSTF, again, are both RNA binding proteins. They recognize that sequence in the RNA and essentially jump from the tail of the polymerase over to the transcript that has produced those signal sequences. The cleavage factors 1 and 2 are associated with these components, and cleavage takes place, cleaving the transcript away from the elongating polymerase. The next step then with the transcript is that the poly-A polymerase adds the poly-A tail to the 3' end. So essentially you get cleavage of the transcript and poly-A's added to the tail of, or the 3' end of the transcript. However, what's missing from what I just described for you is actually terminating transcription. So the polymerase keeps on going. All we've done in this series of steps is essentially cut away the transcript from the elongating polymerase. Okay, so what we described in the last slide was essentially the cutting away of the transcript, the addition of the poly-A tail, but we have not terminated transcription. The polymerase keeps on going. So what we're going to address with this particular figure and slide is how termination of transcription actually does occur. In this figure, in panel A, you see the polyadenylation signal sequence in the gene. There's an additional signal sequence downstream of the poly-A site known as the COTC site. It is short for co-transcriptional cleavage site, and it too is a sequence element. So what happens is the polymerase transcribes through both of these signal sequences. You have the cleavage and the polyadenylation factors, like we just talked about in the last slide, all do their job, basically cut away the transcript from the elongating polymerase. But this polymerase is still elongating or continuing. It hasn't stopped transcription at all. What then happens is we have a self-cleavage event of the COTC site. This portion of the RNA is essentially referred to as a ribozyme, and a ribozyme is an RNA molecule that has catalytic activity. So this sequence has the ability to cut itself. And when that cut occurs, this allows for an additional enzyme known as XRN2 to play a role in this process. XRN2 is a 5' to 3' exonuclease. It essentially jumps on where the cleavage has occurred and starts to degrade the RNA, moving its way closer and closer and closer to the polymerase. Essentially, it gets so close to the polymerase that it torpedoes or pushes the polymerase off of the gene, and hence this is how transcription actually ends.